Hello, my name is Ronaldo de Silva, and today I will talk about cells and its two types. The cell is the basic structural and functional unit of all living things. Every cell, from the simplest to the most complex, works as a miniature autonomous molecular factory. All the necessary activities to keep it alive are carried out frenetically and tirelessly. This is true from the smallest bacterium to any of the cells that make up our organism. Cells can either be prokaryotic or eukaryotic. Each type is different in size and the type of organelles they contain. Prokaryotic cells, meaning before the nucleus, are found only in bacteria and archaea, while eukaryotic cells, meaning true nucleus, form the structure of protists, fungi, plants, and animals. Prokaryotic cells were the first organisms on the planet and are still extraordinarily abundant today. They are characterized for being unicellular and lacking a nucleus and other membrane enclosed organelles. There are two categories of prokaryotic cells, bacteria and archaea. The cells of these two groups are similar in appearance and size, but differ in structure and metabolic pathways. Examples of prokaryotes belonging to the bacteria domain are pathogenic species such as Helicobacter pylori, which can cause stomach ulcers, or Clos- Clostridium botulinum, which causes botulism. Examples of archaea cells include methanogenic organisms, which release methane as a byproduct of obtaining energy. Eukaryotic cells are much more complex than prokaryotic cells, and because they are the fundamental unit of organisms as varied as animals, plants, fungi, and protists, they are highly diverse. The essential difference between prokaryotic and eukaryotic cells is that eukaryotic cells have a nucleus. The nucleus is a structure that stores and preserves the genetic material in the eukaryotic cell and is characterized by being surrounded by a membrane that separates it from the rest of the cytoplasm. The function of the nucleus is to regulate the activities that take place in the cell. Likewise, the nucleus directs protein synthesis through messenger RNA, which is transported to the cytoplasm through the pores of the nuclear envelope. The nuclear envelope has a function of regulating the entry and exit of various proteins and macromolecules.
Hi, I'm Valeria, and I'm going to explain to you the eight characteristics in all living things and the differences between the eukaryotic and prokaryotic cells. First, we all know that there are eight characteristics in all living things. So, number one, they all are made up of cells. They can be unicellular or multicellular. Second, they all have metabolism. Third, they all maintain homeostasis. This is the maintenance of their internal environment, like humidity or temperature. 4. They all respond to stimuli. This is the perception and response to stimuli that come from external and internal environment. Then, they all have genetic code or DNA. Also, they all growth. This is the increase of volume. They all reproduce. They can be in asexual or sexual reproduction. And finally, they all have natural selection. This means that only the ones that can adapt will survive. Now, the differences between the eukaryotic and prokaryotic cells. First, the prokaryotes are always unicellular, while eukaryotes can be unicellular or multicellular. Second, eukaryotic cells are larger than the prokaryotic cells and are much more complex. Third, prokaryotes don't have a nucleus while eukaryotes does. And finally, in eukaryotic cells, the DNA is found in the nucleus, while in prokaryotic cells, the DNA is stored in the cytoplasm. That's it.
Hi, I'm Tamara, and I'm going to talk about the history of the microscope and cell theory. So, in the late 16th century, Hans and Zacharias Janssen made the first compound microscope, which is a microscope with more than one lens. This device made it possible to magnify objects between 3 and 9 times In 1665, Robert Hooke designed a microscope made up of three lenses that was able to magnify samples between 20 and 50 times their actual size. In 1667, Anton van Leeuwenhoek perfected the simple microscope, which is a microscope that had only one lens, allowing him to magnify objects 200 to 300 times their original size. It is vital to consider three concepts when using a microscope: the magnification, that is the ratio of the size of the image of a sample to its actual size; the resolution, that is the minimum distance between two objects, which makes it possible to distinguish them as two independent objects; and the contrast, that is the difference in brightness between an image's light and dark areas. Now, the cell theory, that proposed that all living things are made up of cells, was proposed by Matthias Schleiden, Theodor Swan, and Rudolf Virchow in 1665. Robert Hooke published his work Micrographia, and Leeuwenhoek did so in 1667, describing them as small animalcular. In 1858, Rudolf Virchow carried out a series of research studies on cancerous tissues that allowed him to demonstrate that every cell comes from a previous cell. The cell theory is based on postulates: all living things are made up of cells, it is physiological unit of every living being, and all cells come from another pre-existing cell. The cell contains hereditary material, and disease starts in the cell. So that was everything for this topic.
Hello, my name is Patricia and I'm going to talk about cell transport. Cell transport is movement of materials across cell membranes. First, we have passive transport, that is a type of membrane transport that does not require energy to move substances across cell membrane. Then we have active transport, that is transport of molecules that require the use of energy in the form of a energy storing chemical called ATP, adenosine triphosphate, to help them across the cell membrane. Then we have osmosis, that is a process by which molecules of a solvent tend to pass through a semi-permeable membrane from a less concentrated solution into a more concentrated one, thus equalizing the concentrations on each side of the membrane. Then we also have diffusion, that is defined as the movement of individual molecules of a substance to a semi-permeable barrier from an area of higher concentration to an area of lower concentration. Then we have facilitated diffusion, that is the passive movement of molecules along the concentration gradient. Then we have endocytosis, that is taking in of matter by a living cell by invagination of its membrane to form a vacuole. And finally,
Hi. For this video, I'm going to talk about the levels of organization and homeostasis. But first, I'm going to talk about the levels of organization. Just as a state needs organization, our body is not the exception. Like, have you ever seen the nested Russian dolls by hierarchical sizes? Well, we can say that this is a similar example to how our body is organized. At first, we start from the atoms that are the submicroscopic level of our body. Then the molecules. These are formed from a group of atoms, and then the molecules turns into organelles to build the parts of a cell. From the cell become the tissues. And of these are four types: the connective tissue, the epithelial tissue, the muscle tissue, And the fourth one is the nerve tissue. From the tissues, it builds the organs, which each one having a specific function. And finally, a group of organs forms the systems. For example, the digestive system, the nervous system, skeletal system, and so on. Now, from the homeostasis, the first to propose the concept of body balance was Hippocrates, but it was not until 1929 that the physiologist Walter Cannon named the homeostasis the action of this regularity mechanism. For example, when it's hot or cold or outside, this is we can call this as the stimulus. Then the sensor is our brain, but specifically the hypothalamus, and the effector will be sweating or shiver. And that's the information for my topics. Thank you.
Hi, my name is Anna, and I'm going to talk about three topics. The first one is pregnancy, the second one is addiction, and the third one is obesity. I'm just gonna give a little description of all these topics, but it works. The first one is pregnancy, that is the condition between conception, the fertilization of an egg by a sperm, and during which the fertilized egg develops in the uterus. Your weeks of pregnancy are dated from the first day of your last period. This means that you're in the first 2 weeks you're not really pregnant. Your body is just like preparing for the ovulation. The second topic is addiction. The addiction is defined as not having control over using, taking, or doing something to the point that it could be so harmful for you. Addiction is most commonly associated with drugs, alcohol, smoking, gambling, but it's possible to be addicted to just about work, shopping, solvents, internet, and stuff like that. And the strain of managing an addiction can seriously damage your work, life, and relationships. In the case of a substance misuse like the drug and the alcohol, it can have serious psychological and physical problems or effects. And the obesity is a complex disease involving an excessive amount of a body fat. Obesity isn't just a cosmetic concern, it's a medical problem that increases the risk of other diseases and health problems such as heart disease, diabetes, high blood pressure, and certain cancers. Thank you.